manipulation to have enough information to be able to manipulate biological agents for the wrong purpose. And for us to have a clear understanding of these benefits and risk that goes with artificial intelligence, GETS has decided to organize this platform, this webinar, to bring together top experts in the area of emerging biotechnologies to discuss how the continent of Africa can maximize the benefits from artificial intelligence and at the same time develop policies and rules and governance structures that will prevent the risk that goes with it. And that is why we're bringing two top experts to discuss this very important issue with us today. So without wasting more time, we'll go straight to our speakers. But before we go to our speakers, let me lay down some rules that will guide the webinar today. We're having two speakers with us today, two top experts on the subject matter, and each of them will be speaking for about 25 minutes. After their presentations, we have about 10 to 15 minutes time, 10 to 15 minutes for question and answers and discussion. For our participants that would like to contribute or ask questions, kindly go to the question and answer section of the platform we're using and put in your questions there. We're going to respond. The experts are going to answer your questions after the presentations. Also, to our participants, you can go to our chat box. Let's know you are here. You can put your name and the country you are joining us from. We have an idea of those are the names of those that are joining us and the countries that are joining us from in this webinar. I want to say a very big thank you to our experts for agreeing to be part of this webinar, and also a very big thank you to our participants. Without our participants, this webinar will not be very successful. So thank you to our participants and our speakers for accepting to be part of this webinar. Without wasting our time, we'll go straight to our first speaker. Our first speaker today is Dr. Sarah Alcata, and I'm going to be introducing her briefly and hand over the floor to her to speak. Dr. Carter is a principal at Science Policy Consulting LLC for over 15 years. She has focused on advanced biotechnology tools and capabilities, biosecurity screening frameworks, and international norms for biosecurity. In recent years, she has supported the Nuclear Threat Initiative, NTI, on projects related to DNA synthesis, screening, and the implication of artificial intelligence for biosecurity. She's also a senior fellow of the Federation of American Scientists. Previously, she worked with policy centers of the J. Craig Venter Institute and at the White House Office of Science and Technology Policy. She's a former AAAS Science and Technology Policy Fellow and a former Messiaen Science and Technology Fellow of the National Academies. She earned a PhD from UCSF and a bachelor's degree from Duke University. Dr. Sarah, we're very happy to have you speak to us on this platform today. Please, you have the platform to speak for about 25 minutes. Thank you.
Great, thank you so much, Dr. Bobodoye. And this is a very important topic. I'm very excited to be here with you all today. I will say that I have been working with NTI, the Nuclear Threat Initiative, for more than 5 years now on these topics. And only in the past 2 years has AI become a huge, topic. It sort of exploded onto the scene and everybody started talking about the intersections of AI with biology and what those implications would be for biosecurity, both the benefits and the risks that they might pose. I am going to talk today about a report that I helped to lead with NTI. Here, I'll share my screen. I hope that works. Let me pull up here, let me make sure I have the. So about a year ago, we decided that NTI decided to pursue this project, the convergence of AI with the life sciences. And we were going to focus on both the benefits and the risks, but the risks that we were talking about were a little bit narrow. They were very much focused on the risks, the biological harms that could come from misusing tools of AI. So I'm going to talk a little bit about that report and what we did to get to this report and some of our conclusions. So for this talk, I'm going to be talking first about the research process for this report, and then next I'll talk about the key findings and then the recommendations that we at NTI decided to put forward so that we could mitigate risks while still claiming some of the benefits. And then some of the next steps. This report was launched in October this past fall, so the next steps are things that we at NTI are pursuing now to try to implement some of the recommendations that we decided were priorities. So for the research process, the key questions that we wanted to focus on were first, what are the current and anticipated capabilities for engineering biology? So this is a little bit narrow, you'll see, about the scope of things that the scope of impacts that AI will have in the life sciences. We were really focused on engineering biology. I know that my other panelist, Dr. Sengupta, will talk a little bit more about some of the broader implications of AI for biology. And then our second question was, what are the biosecurity implications of these developments, both the benefits and the risk of misuse of these capabilities? And then third, what are the most promising options for governing these capabilities so that we can reduce the risks while still maintaining the benefits? So last summer, we were doing interviews. We did it with many different experts. These experts included experts in artificial intelligence, experts in engineering biology, experts in biosecurity and biosecurity risk mitigation frameworks. And we asked them all about questions about those key questions that I just mentioned. We held a workshop last August with some of those same experts to talk about some of our preliminary findings. And then once we had written up a first draft of the report, we sent it for a broad peer review to make sure that our findings represented the understanding of all of those experts. Here's some of the key findings that we found. So we decided, after speaking with all of these experts, we determined that we needed to look at 3 different types of artificial intelligence models. The first sort of set that I'll talk about are large language models, which are the kind of the largest kind of ChatGPT type AI models. The next is AI, what we called AI biodesign tools. So these are much more narrowly scoped biodesign tools that are trained on biological data, and I'll talk a little bit more about those. And then the third type of AI that we talked about was automated science, so just the ability of AI to automate the scientific process. So first on large language models. So this is, these models are basically, they're trained on large amounts of data. You know, these days, some of these models are trained on the entirety of the internet or, you know, huge amounts of human language. There are large language models that are trained in English, for example, and all of the English language content that they can get a hold of. There are also large language models trained in other kinds of languages, for example, Chinese, or other natural languages. And for the capabilities, when we talk to our experts, what they thought that the intersection of these large language models with biology and with engineering biology would be that they could function as teachers and assistants. You know, they could help you troubleshoot. If you were in the lab, for example, and we're trying to engineer biology, you could ask a ChatGPT or a similar large language model, you know, what am I doing wrong here? And it would be able to help you figure that out. And it could also help in training. So if you had an undergraduate student or you know, a young person who is trying to get up to speed quickly on a particular topic, it could really help bring together lots of information and help that person better understand that topic much more quickly than with older methods of training. However, these large language models, because they are trained on existing information and you know, things that they can find publicly available, they have a limited ability to generate novel insights. So especially, so in the laboratory, for example, they would be most helpful for experiments that were very well documented, where many people had put information about how the experiment was supposed to go. It would be very difficult for them to have a new idea about how to proceed in the lab. Some of the biosecurity implications of these tools, the sort of the risk side, is that they could lower barriers to misusing biology. So in the same ways that they can help somebody trying to do good in the lab, they could also help somebody who is trying to do bad and trying to misuse those capabilities. However, the experts we talked to were not very worried that large language models and ChatGPT type models by themselves would really be very empowering to a bad guy trying to misuse biology, for example, to create a new pathogen. They thought that there were way too many, barriers still remaining. They thought that it would be very difficult for these tools to really empower somebody who is trying to cause trouble in this way. The next category is biodesign tools, and these are tools that are much more narrow in scope. They are trained on biological data, for example, DNA, RNA, protein sequence, or even more complicated types of biology. For example, scale up data when you're trying to scale up a microbe into a bioreactor. These are things that are much more specific to biology and the biological properties of your system. For these capabilities, by far the most developed types of tools are for protein design. So some of you may have heard of a AI model called AlphaFold, and this was a protein model that came out in 2020 that was able to predict protein sequence from the DNA sequence very reliably, much better than any previous tools had been able to do so. And so and protein design has continued to advance very rapidly. And for biodesign in general, that excels in well-defined problems with plenty of data. So for example, protein design was able to advance very quickly because there was a lot of data. Scientists had spent a lot of time curating databases of structure and function, or of DNA sequence and structure and functions of those proteins. So there was good data for the biodesign tool to be trained on. The biosecurity implications of these, I'm actually going to get to some of the benefits in a minute, more of the benefits of especially the protein design tools in just a minute. But on the risk side, the biosecurity implications of these, our experts were much more concerned about these types of tools. In particular, because they could potentially generate novel insights. They could generate, they may be able to produce protein sequences, for example, that are not found in nature, but could potentially, as part of a virus or something, cause extra harm, cause more harm, or harm in a way that is not found in nature. And that was something there's a lot of uncertainty about that, but there is a concern among the expert community that these biodesign tools could be misused in that way. I will say that engineering complex traits in pathogens remains extremely difficult. So it's because of the issue I just mentioned, there's not a lot of data. That's very hard to predict what a single point mutation that will dramatically increase transmissibility in a virus, for example. And so because there's very little data about that, it's very hard to have a good AI tool that can make that kind of prediction. One of the more near-term and certain biosecurity risks related to biodesign tools is that it could allow a bad guy trying to misuse biology to evade DNA synthesis screening. Some of you may know that many DNA providers around the world, they will screen the orders that they get. Commercial providers will screen the orders to see if an ordered sequence of DNA matches pathogen or toxin DNA. And if it does match, then they'll follow up with the customer to make sure that it's being used responsibly. But some of these protein design tools could change the sequence enough that it has no match with what's known to exist already, and therefore these screening frameworks are going to be ineffective. The last category of AI that we talked about was automated science. And some of the capabilities, what we're talking about here is is automating steps in the scientific process. And this goes all the way from doing background research to generating a hypothesis that you might want to test experimentally. It also means integrating these AI tools with robotics. And automated laboratories and generating the data, analyzing the data, even writing up the report in the end, the paper that might be published. And right now, these capabilities are very limited. Each step in this process is actually being automated, and there are AI tools being generated to try to do each of these things, but they're very limited right now. And the laboratory robotics in particular are very limited to very simple experiments, things that are very well described, easy to do. So they're not going to be very powerful in the very near term. One of the biosecurity risks implications of this is that these types of automated science could facilitate the interface of AI agents with other tools. For example, it could make it much easier to use a biodesign tool or a protein design tool by linking a large language model with that tool. Right now, many of the biodesign tools are very difficult to use. They require a lot of expertise. They're definitely made for scientists, but by using large language models in combination with those kinds of tools, it could make it much easier to get those kinds of novel designs. All right, so now a little bit on the benefits. So the big categories of benefits are there's improved development of vaccines and treatments. There could be improved DNA synthesis screening. As I mentioned, this is the framework that allows commercial providers of synthetic DNA to detect if somebody is trying to misuse pathogen or toxin DNA. And also improved detection and attribution when there is an outbreak or when there is a biosecurity or a pathogen concern in the community. Some of the things that we've heard from our protein design colleagues is that these types of tools, especially the protein design tools, are already starting to contribute to development of vaccines and other medical countermeasures. For example, at CEPI, they have a 100 days mission where the idea is that they will want to, whenever there's a new pandemic pathogen that comes up, they want to have a vaccine ready in 100 days. And for that to happen, they are already relying heavily on AI tools to identify candidate vaccines much faster. And they're already putting money into some of these tools. They're putting in money into sort of more automated ways to get to a real vaccine. In the future, AI could also help automate more of the process to detect a bad outbreak, to develop a vaccine candidate, to test those candidates, and to help even in the sort of manufacturing of those vaccines. And one other benefit that I would just want to highlight is support for scientific research and development. I mentioned that especially the chat bots, the kind of large language models, really can help with training and troubleshooting. So helping people come up to speed on some of these very complex topics. And also large language models could make the biodesign tools easier to use. So I've sort of said that in a negative way about the risks, but really these tools together could really help unlock a lot of these tools that have been developed in other contexts in the global north, for example. These large language models could make them easier and more accessible for a broader range of people. I'm going to talk about opportunities for risk reduction very briefly here. When we talked to our experts to develop this report, they had all kinds of ideas for guardrails for what you could do with an AI model to make it safer. And all along the life cycle of model development, the model development process. And some of these, we talk about guardrails because we do want the AI models to proceed because the benefits are too much. You know, they're too big to want to give up. But the guardrails can kind of can help keep them on track. So some of these ideas were controlling access to training data. You could control access to the computing infrastructure needed to actually train these models. One of the big ones that has been coming up a lot is model evaluations, especially for these chatbots and things, trying to figure out what the risks really are. How powerful is this tool? How much will it, how much information will ChatGPT give you about developing a bioweapon, for example? And there's a lot of discussion about how to do that. And then obviously in the sort of final stage, you could control access to the model and not let people use it unless they have a good reason to. But all of those will have to be, all of these guardrails need to be sort of further refined to make sure that we can harness the benefits. Obviously, and that's not the opportunities for risk reduction are not just on the model. The other things that we came up with were first to bolster biosecurity at what we call the digital physical interface. So that's where the digital designs that exist in a computer become actual biological reality. So a couple of the ideas there were to strengthen DNA synthesis screening and expand and improve customer screening to make sure that we can, that people are not trying to misuse these tools. And then obviously foundational to all of this is to strengthen pandemic preparedness. So bolster existing systems and even integrate a lot of new tools, AI-enabled tools to improve pandemic preparedness and response. So the recommendations that we came up with, I'll just go through very quickly. They fell into three buckets. One is on AI biogovernance. Here, I'm just going to highlight an international AI bio forum that I'll mention in more detail in a minute. AI bio safeguards, these are the guardrails that I talked about. Some of them are pretty promising, but others need a lot more research to figure out what will be a good idea for these. And then there's on the biosecurity and preparedness, it's some of the same things that I was just talking about, strengthening biosecurity at the digital physical interface and integrating new AI tools into pandemic preparedness and response. So the next steps for us, I just mentioned the International AI Bio Forum. So NTI is kicking off an International AI Bio Forum. The first meeting will be in April of this year. Some of the key things that we hope for this AI Bio Forum is that it will be a place where people around the world, many different kinds of stakeholders, very international focus, can come together to talk about best practices for AI bio guardrails. Some of the early discussion points we believe will be about model evaluations to better understand risk, and also managing access to models and how to do that fairly, equitably, and so that people can still get to these models to make sure that we can get their benefits. And I just put this on the bottom, establishing international norms for AI biogovernance. I do want to emphasize the international piece here. Almost all of the discussion that we have heard on these topics has happened in the context of the US and the UK, and that's we are acutely aware that this does not represent all of the stakeholders of the world, and we are trying very hard to make an international forum where we can make sure that the approaches that we develop are appropriate for the whole world and that we can all move forward together. So this is going to be a very important piece of this effort moving forward. And then the last thing I'll mention is just that we are working on ideas for developing guardrails for biodesign tools. There's been some discussion about large language models, but much less discussion about biodesign tools. For these tools, many of them are open source, so they're available to everybody. Many of them were developed in large collaborations. They're very academic. They are, widely available, including all of the source code and everything. But we're trying to explore options for managed access to these things. And we're going to be talking to some stakeholders, trying to talk to different kinds of people to see if there are options for restricting access in a way that can be equitable and that can ensure access to the people that need it. But as you can imagine, this is a very difficult question because we do want to make sure that we can get to the benefits of these tools. And we'll be conducting some stakeholder outreach and community building to help talk about those topics. With that, I will wrap up. I just want to say thank you. You can contact us. My email address is here, carter@sciencepolicyconsulting.com. I will put that in the chat as well, because I'd be very interested to hear any of your thoughts about it. I also want to, Jamie, to acknowledge the other leaders of this report. Jamie Yassef is her email address is here. She led the report from the NTI side. And also my colleague, Nicole Wheeler at the University of Birmingham. She was the other lead author on this report. So with that, I will take down my slides. I'd be interested to hear any questions or comments you have about it.
Yeah, thank you, Dr. Sarah, for that very detailed and interesting presentation on the projects that you are involved with NTI. I want to say that Dr. Sarah and NTI, they've been at the forefront of artificial intelligence and biosecurity in the world. Like she said, most of the activities is based on activities going on in the United States of America and the UK, and a whole lot of work has been done around preventing misuse of artificial intelligence and other forms of emerging technologies and using them for their own biosecurity purpose. So thank you for the great work you're doing, and hopefully we'll, like you said, we hope there will be a global governance system around AI and other emerging biotechnologies, because the truth is that the world has become a global village, and whatsoever biosecurity threats we have anywhere in the world can become a global issue within a short period of time. So it's very important for us to start having global view of most of these possible threats and having global collaboration to effectively address most of these threats. Yeah, Dr. Sarah spoke about a whole lot of things about some emerging artificial intelligence tools and their benefits and their risk. She talked about the projects that she was involved in, where about over 30 experts were interviewed, workshops were conducted, and she talked about the main AI models that they focused on, the large language models, the LLM, the AI about design and tools, and the automated science. And LLM, and she talked about the benefits and the risks that goes with them. Like, for example, LLM can be used as a teacher, a coach, to give direction to basic information about science and biology and activities in the laboratory. But it has not been developed to a stage that can be used for currently for developing high-tech issues around development of biological weapons and things like that. But nobody knows what will happen in the future. You know, most of these LLM models are improving on a daily basis, and we just have to guide the kind of ChatGPT that will be released in the nearest future to make sure that it doesn't go to a level that it can provide information for non-experts or junior experts to develop bioweapon or manipulate biological agents for the wrong purposes. She talked about biodesign too, their capacity, their capabilities, and biosecurity implications, and also automated science, identifying their capabilities and biosecurity implications. She talked about the AI biogovernance and the workshop that will be coming up in April. We have been invited to that workshop and definitely get to be part of it. And the importance of developing a global governance structure that will govern AI and some other emerging biotechnologies to make sure that we start having a global view or a global risk identification and prevention strategies for emerging biotechnologies in the world. So thank you very much, Dr. Sarah, for your very detailed presentation. And I want to our participants, I know you have a lot of questions. for Dr. Syria, just go straight to our question and answer to his question and answer section. And type your questions there. We're going to take your questions after the second presenter. We will not allow, I can see some participants raising their hands. We're not going to allow participants to contribute for now, but after the presentation, after the second presentation, we can give the opportunity to one or two participants to speak and also take the questions from the question and answer section. So without wasting our time, we'll go straight to our second speaker. Our second speaker today is Dr. Aparupa Sengupta. I hope I pronounce the name well. Dr. Sengupta is an accomplished scientist and a global life science security practitioner with more than 15 years of experience across the field of infectious disease containment and global biological risk reduction. During the COVID-19 pandemic, she was named a COVID hero by her then employer, University of California, Merced, for her invaluable services in helping the community steer through the pandemic crisis. At NTI BIO, she leads the Biosecurity Innovation and Risk Reduction Initiative with a goal of promoting responsible development and use of biotechnology and emerging technologies while safeguarding the risk. Dr. Sengupta is a highly sought-after speaker and expert in the global biological threat reduction sector and has given numerous talks advocating for the safe and secure use of emerging technologies such as CRISPR-Cas9 and other emerging technologies. Dr. Sengupta is originally from India. She holds a doctorate degree in biological sciences and a master's degree in molecular genetics and biotechnology from Michigan Technology University and an MSc in biotechnology from Bangalore University in India. Dr. Sengupta will be speaking on artificial intelligence in life sciences, navigating risk and rewards in Africa and Global South. Dr. Sengupta, thank you for joining us. You have the floor, please.
Thank you so much, Dr. Bobadoy and the GET Consortium for having us here, having me here. And I'd like to thank my fellow panelist and my colleague, Dr. Sarah Carter, for setting the stage so beautifully. She set the stage perfectly for me to take on and kind of move the lens a little bit towards Global South and talk about the opportunities and risks there. Before I share my presentation slide, I would like to say that for today's presentation, the views and opinions that I'll express are my own and are not that of my organization. And without further ado, I will share my slides. Are my slides visible to everybody? I hope so. Okay. So Dr. Carter already set the stage by talking about what artificial intelligence is and how it is an extremely important topic, clearly, across the globe, and what kind of progress and what kind of opportunities that there are. And of course, there are also underlying risks. I'm going to talk about generally a bunch of them, as Dr. Carter mentioned, but I will keep my focus towards the rewards and the risk in Africa specifically and in the Global South. So before I dive into my presentation, this is just to provide an overview. We'll talk about the AI opportunities in life sciences in general and the risk associated with adoption of AI technologies in Africa and Global South. Then we'll talk about the current challenges, and of course, we'll try to look into the future challenges as well as this AI capabilities keep developing. We'll talk about recommended mitigation strategies and the implications side as well, especially on the biosecurity side. And then we'll talk about the future of artificial intelligence in Africa and how we can maximize the benefit for a better future. So we'll start with the AI opportunities in life sciences. Keeping in mind, if you think about if we take a couple steps back and if you think about the bigger picture here, some of the challenges that global